used with these things called organoids.
Oh, yeah.
What are those exactly, and how are they helping scientists study diseases? So organoids are pretty fascinating. Imagine you could grow tiny 3D versions of your organs in a lab, like mini organs in a dish.
Oh,
That's essentially what organoids are. They're small clusters of cells that scientists grow in a special way so that they organize themselves to kind of mimic the structure and function of a real organ, like your liver, your brain, your intestines. It's like creating a tiny model of the real thing.
okay, so mini organs in a dish. That already sounds like something out of a sci-fi movie. Why are these things so useful for studying diseases, and how does CRISPR fit into all of this?
Well, the great thing about organoids is that they behave a lot like the real organs in your body, but they're much easier to study because they're right there in a dish. You can poke and prod them and see what happens. So let's say you want to understand a disease like cystic fibrosis.
Okay.
Cystic fibrosis is caused by a problem in a specific gene, and scientists can use CRISPR to actually introduce that exact same genetic problem into a healthy lung organoid that they've grown in the lab.
So they're basically recreating the disease in a dish.
Exactly. They now have a model of the disease right there in front of them that they can study up close.
Wow.
It's amazing.
So instead of trying to study the disease in a whole person or an animal, they can study it in this tiny lab-grown version. That's a game changer.
Oh, absolutely. And they can do the reverse, too.
What do you mean?
Well, they've taken organoids that were grown